What's been happening with you guys?
Nothing. It's boring at the moment. There's not much happening in long, dark days. Don't like this time of year.
Oh, we've got a new face. Ed basically rang me a couple of days ago. I didn't have time to talk to him. Say hi.
Hi there, Daniel. How's it going?
Yeah, very well.
Good, good.
Everybody here knows each other, so why don't you sort of, introduce yourself?
Okay. My name is Ed Reynolds. I'm an arable farmer from Cambridgeshire, and yeah, I'm currently following some of the conservation agriculture practices on my farm, and I saw Daniel's presentation in the Oxford Real Farming Conference, and I thought, yes, this is the person to talk to, because I've got a few questions with regard to yeah, specifically compost and the application of good biology into my growing system, which is obviously a massive topic. And yeah, I've made a start, but I'm really interested to get a few answers to some specific questions.
Yeah, you can go for it if you want.
It must have gone quite well then, if your talk attracted new people to the group.
There you go.
Well done.
Well, thank you very much.
Fire away, Ed. What are your questions?
My question. So I've got a couple. I've basically got a Johnson Sioux bioreactor, or several of them, which are coming to the point of maturity, as you might say now, and I'm not entirely sure how to get them involved in a sort of cereal growing scenario. What the best sort of way of applying it to the crop, or to the seed, or to the soil, or whatever it is. And I just wanted to ask somebody who's had actually some experience of trying to get the all the goodness out of the reactors and into some sort of liquid or something.
Yeah, there's a couple of people that they're not actually here, but there's a couple of people with some good experience in this group as well. I'm thinking of Tommy, Andrew, John, but we've also obviously been doing our own applications, and you can do, like, the simplest thing obviously is to spread the compost, but then you're risking it drying out on the surface. So what we do basically is we try and well, we call it drenching the soil, but really on this application it's more of a mist. But basically what the ideal scenario is, you want to extract the microbes into water, and then you want to inject it in the soil before the crop. So that's the first thing that you would want to do. And so you probably would need to look into modifying the equipment if you can. So you would need some sort of a ripper basically that would allow you to sort of drip behind the rib, so that you can go beneath the compaction layers that you've got. And that would be sort of like the ideal way of doing that. And then the second way is to have it whilst you're seeding. And so the easiest things that we've seen so far is basically when people have got a tank on the front of the tractor, a pump, and a distributor that goes in front of the actual drill. And so you've got a little drip that basically sort of follows the seed, kind of thing. And so in terms of extracting, you need to, the easiest way that we basically found was to put compost into water and then agitate it using a paddle mixer. And then once you agitate it, you've basically got these microbes in the water. Then you need to take out the bulk of the material, and you do that with these pond filters. So that's basically a 400 micron opening bag, nylon bag. And this basically gives you now a suspension of microbes and some organic matter of particle size no greater than 400 micron. And then what you need is, if whatever machine you're using, basically you want to have a filter no smaller than 600 micron, and that basically gives you a relative guarantee that you're not going to get clogged on the first pass. So they still can get clogged basically with these particle sizes, but it's less possible with a decent size. And then so then in terms of the injecting behind the tines, usually you don't need a filter, so it's usually pretty straightforward. There's really nothing to clog. And then if you're doing the same thing with the seed, then you don't need to worry too much. There usually isn't there's any filters in the system. And then we have seen here actually a way of applying to seed before the seed goes into hopper. We actually still haven't had a good success with applying to seed, mainly because we're greedy and we just want to put so much in the seed. Our friend from Ireland, Tommy, he basically he had a little dribble of an extract onto a seed going into the hopper, basically. I can probably find it, but I'm just going to finish the sentence. He's basically applying a very, very small amount of the extract onto the seed before it goes to hopper. So the seed isn't really wet. It's just got a little bit of dampness to it. So the two main applications you're looking at, basically applying to the soil and applying onto the seed. And again, the least troublesome are the ones that require the most changes in terms of the equipment. Sorry. And then we would basically then follow it with at least 3 applications to the plants. And so we would basically brew a tea from that material. So we would take that material and apart from doing an extract, because we would still do an extract, we would also brew it for 48 hours with a good pump. We use pumps, we use IBC tanks, and we use pumps which basically give you 275 liters a minute of air movement. And we measured the oxygen levels in it, and it's like 11 parts per million, which is like twice of what you need not to go anaerobic. So this is enough. These are pumps for sort of hydroponics, aquariums. You can even get them on eBay. I bought them on eBay or on Amazon. So the thing is that in general, we have used about 70 liters of our compost per application per hectare. Now the application rates, there you go. This is your perfect setup. If you look up right now, Ed.
Yep.
This is basically Adam is showing you like a perfect setup. This is a cone tank. It's got a split in the bottom, so you've got air line coming in, and there's a valve below the air line, which means that it allows you to close off the whole tank, while the air can go in. And then basically once you're done pumping, then you close the air line and you open the dump valve at the bottom. And we've got like a straight through valve with like a 1-inch opening in this particular one. This is just a 200-liter tank, so it's not a massive one. We use this for our brews. But we're not on a very, very large scale yet, so we don't have to have very massive tanks. But you can get cone tanks, which are like 4,000 liters, so you can get quite a lot of quite a big brew going. But we need a big pump as well for that. So yeah, we basically use about 70 liters of compost per hectare, extracted, and then some of it we'll be brewing as well. And that has given us decent numbers, in the actual volume of the water of microbes. So we do check under the microscope, what is the actual result of our work. People have gone as little as 2 liters of compost, because this is like the minimum that Dr. Johnson has sort of demonstrated. Now, we haven't done 2 liters. We are impatient. And so this is why we're basically just load the extract as much as we can. And we had good results with that, in a sense that we've got a 5-hectare trial right now, and we have physically seen microbes establish in a matter of weeks. And so this is what gives us confidence that, if we increase that volume basically of material that we use up front, then we actually have a chance of establishing these guys in a relatively short time. Now, in terms of the results for that, you'll have to wait until August. I'm sorry.
Very interesting. Okay, thank you. There was a lot of information there.
All at once. Mind blown. Sorry.
No, that's really interesting.
What are you growing on your farm? Mainly cereals.
Yeah, cereals. So wheat, barley, oats, and then we have linseed, oilseed rape, and flax, and beans, peas.
Okay.
So yeah, quite a mix.
So are you, I can't remember from the email, are you certified organic or conventional or?
No, we are conventional. Yeah.
Mhm.
But I was fascinated to hear, various talks like, Elaine Ingham's with regard to the soil food web and how you can develop it and get the balance of bacteria and fungi towards a more workable level.
Mhm.
And there's a potential for everything to change. And it's just, fascinating to hear about that.
So how, and you were telling me what, you've got, did you say 300 hectares? What was that?
Yeah, about that. Yeah.
Yeah, so then how many of these Johnson Sues have you got, did you say?
Just 2.
Just 2. Okay.
Yeah.
So if we were to do our applications, basically you've got about 10 hectares worth of applications in those 2. So that gives you a trial, and you can basically trial it on 10 hectares and you don't risk too much then.
Yes. Yeah. Okay.
So you could literally take 10 hectares, if you can do the calculation, if you can do the maths, right? You could take out those 10 hectares out of your revenue,
Mm-hmm.
and just basically not put any chemicals on it, literally, and just basically trial that. But it's, it's basically up to you, but what sort of size you can actually afford. And then based on that, you can run a trial on that with pretty good concentration of applications. And
Right. Okay.
And when if you need help along the line, obviously you know where to find us now.
There's some other stuff you can make that's pretty cheap as well to support that. So
Okay.
it's worth mentioning.
You need to ask questions to Adam, because Adam doesn't speak much, but when he speaks, you then get a lot of information.
Like what, Adam? Please, more.
So at the moment we're trialing different stuff of biostimulants, and one of these things is my local farm shop gets deliveries from Riverford Organic. Some of that produce isn't it's not good enough for display purposes. It's got like some kind of stupid mark on it that means they throw it in a skip. So I take that out of the skip and I'm trying to focus on stuff like carrots, parsnips, turnips, beetroots, potatoes, anything with like a root structure that's probably got lots of exudate like stuff in it, similar to what plants exude into the soil to feed microbiology. So then you take a good sugar, a dry sugar, because it's dry, it's a crystal. You put it next to the plant tissue and you massage it in or beat it in with some kind of stick or a sledgehammer. And then that builds up osmotic pressure and it pulls the sap across the cell walls without too much, destabilization or like denaturing of the stuff. It's not like a heated process or anything. You're not using high amounts of pressure. You're just using sugar. So then you let that ferment, and you can ferment it for a week or anywhere up to a month. You put a material cover on it so it can breathe, and then you eventually extract off the solids. You can use that same 400 micron mesh that was mentioned to make your compost extract, and then you have like a very easily soluble biostimulant made from these waste products. And we find that's really good for stimulating flagellates, especially whether it's in a terrestrial-like environment or an aquatic environment. The flagellates are really supported, and flagellates can cycle a lot of N, so they can potentially save you on your nitrogen inputs.
How well with peelings, Adam?
What's that, sorry?
Would that work with peelings, like potato peelings?
It will work with anything. It will work with you and me.
Mm.
You could smash me up and mix me with sugar and ferment me,
I think that's a little extreme.
Yeah, so you can get really creative with that process.
This is the termination strategy for all employees in this company. Sorry.
Yeah, that's where they go, into the fermentation buckets. So you can do that with fish. Sorry, Phil, what was that?
I was going to say, you don't feed them to pigs, you feed them to microbes. That's like the
But
old
in natural
school
farming,
way
they
of
do
getting
put
rid of
these
a body.
inputs in the animal water too.
Yeah.
So they do feed it to pigs.
All right. Okay.
But we won't. We just use it as a biostimulant.
So is that like a KNF or JADAM or something? Is that where
Yeah,
that?
it's like the FPJ process, or the fermented plant juice, the fermented
Yeah.
fruit juice process, but I'm targeting like root structures.
Okay. Yeah.
Yeah.
Fine.
Because my theory is, you know, these root structures will grow in the soil next to the environment where these later successional soil dwelling fungi will live. And therefore those soil dwelling fungi are probably stimulated by things that are contained within those root structures.
Yeah.
And you just, I suppose you're taking sort of root vegetables simply because that's the easiest way to get roots, really.
Yeah, and it's free for us, you know.
Yeah.
It's just a resource we have access to, and it's organic, and it works really well. Using the microscope,
Yeah.
it show, you know, it's better than molasses alone.
Yeah. And are you because obviously FPJ generally they like to stick to one type of food source. Are you mixing that up and just doing like a
Yeah, just
all things
mixing it all together. Yeah, just whatever we get. It's got, yeah, like I said, carrot, parsnip, turnip. It's got a lot of diversity. It's got some squash in there. It's got some apples. Yeah, there's a few bananas in there, but it's mainly roots.
Because I kind of figured, like with the Korean natural farming stuff, they never like diversity. Everything's always done in the one of this. Like I always think diversity is maybe a good thing, isn't it?
Yeah, I mean, for us, it would just require me making loads of little like fermentations instead of one big one. So I just threw the dogma out the window and just put everything into one pot. I actually made one with brassicas that's separate. So any waste cauliflower, broccoli, stuff like that, I did keep that one separate just to check it out individually. And then I also made one with fish waste.
Was you thinking with the brassicas that's one that doesn't necessarily form mycorrhizal associations, so might be targeting different fungi? Or
I didn't want to mix it together because I didn't really know at the time, but now I've just got the soil report back. I know that our field is deficient in sulfur, and brassicas are usually really high in sulfur. So I can try and use fulvic acid to carry that biological form of sulfur into the plant tissue in our kind of top-down spray scenario that we're doing.
Okay.
But I don't know if we're going to get a chance to do that this time, unfortunately, but that's how I would utilize it now.
Yeah.
And it contains lots of amino acids and stuff, good stuff.
Yeah. Cool.
And what kind of rates do you need for application, like for brassicas?
We just use 1 ml per litre of water. So, you know, 1,000 litres might go out to our test site, and we put 1 litre in 1,000 litres.
So what sort of area would you be spraying that 1,000 litres on?
Well, we didn't even use 1,000 last time, did we, Daniel? I can't remember. Was it 800? Something like that. And that.
What, per hectare?
I'm talking of when we applied to our biological area. How big is that?
5 hectares.
5 hectares, and we used about 800 litres, did we? Of inoculant.
How much was it?
I think it was
just
800.
one tank. Was it just one tank, basically?
Yeah, and it wasn't quite full, so it was about 800 to 900 litres.
And then he put it into some more water, didn't he?
He probably diluted it a bit, yeah. I can't remember now. It's gone.
Doesn't take a lot though, does it?
No, and it's just a biostimulant to boost those flagellates. You can use it at the beginning of a compost tea to as like a food resource, and then you could probably add another ml as the solution went to the field as a biostimulant out to the field. And then there's another process we've just started, and Phil, you sent me some Ingoldium fungi. Can you imagine?
Yeah, it's pretty crazy, wasn't it?
Yeah, he just went first one. Well, you took 2, and one of them I got 5 drops in, and it had Ingoldium fungi in it. So This is another experiment we're doing at the moment, and we're going to use that in a mineralization tank, which is like a compost tea, but it goes for 1 to 4 months. And we put in some kind of inoculant. So you can use compost, you could use lactic acid bacteria. And I'm trying to see if you could use Ingoldian fungi, which are aquatic fungi from river systems that are high oxygen. And they can offer their services in decomposing wood and leaves and complex stuff like waste streams, like cake meals, seaweed, stuff like this. Any kind of crop residue, possibly manures. Well, definitely manures. And in aquaponics, they use to mineralize fish waste to add back to the system.
Is it the Ingoldian fungi that mineralize stuff? In
aquaponics they just use stuff like lactic acid bacteria, and then there'll be other guys in that mix. There'll be naturally probably some ciliates and flagellates, and they might add some liquid IMO or something like that, and that contains bacterial feeding nematodes, and all of those things are going to eat the bacterial biomass and poop out plant available nutrients. And over the course of a month to 3 months, that's going to build up in the water, just free in the water. And because you're in a closed system, nothing's really changing. It's just resources are steadily being depleted, and but it takes a long time. So I've just put in 1 kilogram of tiny wood fines and 1 kilogram of hemp cake meal, along with 3.5 kilograms of our best compost. And then tomorrow, after it's bubbled for about 24 hours, I'll add the Ingoldian fungi that Phil sent us.
But they should just help the nutrient cycling process as well, huh?
They should just be more evolved at cycling more complex substances.
Woody materials and stuff, yeah.
Than just bacteria, yeah, absolutely.
Is this on the screen? Is this from the sample?
That's from what you sent, yeah. This is basically Adam just jumping with joy yesterday.
From this bottle here, to recycle me on it.
Yeah,
I like that you did. And it had a few little seeds in it and a little bit of foam. So I tried to focus on taking the foam out, and I found it in the foam. The Highland Spring bottle didn't have the fungi.
Yeah, I was thinking the issue with that one, I think the water would have been flowing too quickly there.
Yeah, it looked too clean. It looked it was like I could drink it, whereas the other one had a bit of a discoloration, just a little bit, and had some kind of seed present, some bits of leaf or something. So perfect environment for Ingoldian fungi, apparently. Yeah, or it might be Hyphomycete. I'm not too sure about the specific classification of that thing, but
there was one structure basically that you saw in there where there was these 3 bits that were butted against each other. That then we looked at the paper, about these fungi, and one of the drawings basically resembled that structure, and so this is what gives us some confidence that this is the fungi that you were looking for, basically.
Yeah, was it just the one species you think was in there, or maybe some more?
It's hard to tell, isn't it?
Yeah, we're not specialists on that stuff, so we just have to keep looking. Eventually we might learn something.
Yeah, I mean, down the line, I've just got off the call with Sheffield, and we're going to have a lot of our material actually tested for DNA analysis. So we're actually going to have some answers based on that, what sort of diversity we have, like in terms of species and whatnot. So hopefully in the future we'll have answers like that.
Yeah, I was I read that paper that you were talking about last time, the Ingoldian one. I thought that winter might not be a great time for finding them, but apparently winter and spring are the best.
Yeah, people seem to have this this perspective that in winter everything stops, but in my Johnson Sioux outside, nothing really stops. Maybe the nematodes just go a bit deeper into the pile. So if you want some more nematodes in your inoculum, you might just have to dig a bit more if you want to access them during winter.
Okay.
I'm going to change the subject with a question for Ed. Did you record what you put into your Johnson Sioux?
Yes.
Okay.
Yeah.
Because that's interesting, you know, because Dr. Johnson, you know, he's on record to say that it doesn't matter what you put in there. But I'm just very curious, you know, so like to gather this information. We record everything for all of our piles. So now we're getting to the point where we're going to have this DNA analysis done. We'll be able to start cross-referencing these things. So that's going to be, you know, that's interesting, basically from the perspective of, you know, like understanding really, okay, it's great. You know, we all got excited about this whole message from Dr. Ingham. Now let's actually start looking deeper into this subject. So it was funny actually, because, you know, I started this about 5 years ago, and, you know, even back then, Dr. Ingham would say, you know, don't worry about the species level, because, you know, leave that to your children and grandchildren. But she forgets that she's been at this for 4 decades. So I think it's time now that we start looking at these things. So it's going to be interesting to start gathering that sort of data. So.
Can I ask, one of the other things I want to do is try and turn, I take 1,250 tons of cattle manure every year and spread it as manure, but I want to try and turn it into compost. And I think I've got the contractors organized to be able to aerate the pile.
What are you using?
Just one of those tractor towed spinners that go over a pile and actually aerate it. But would it be possible to use the material, the compost from the Johnson Sioux bioreactors, to inoculate that? Do you need to add anything, or is it sufficient to just aerate the manure pile? Or do I need to add a third material in bulk that has a higher carbon to nitrogen ratio?
Yeah, you're probably spot on that last one, because the thing is that majority of people who work with manure, they usually sort of like end up with the bedding, which just has, you know, so like 30-50%, you know, of manure in the thing, or and they end up with a salty material with a high pH, and I probably think that they don't turn it enough as well. So you do want to have more woody materials in there, because even at a small scale that we are at, where basically we do everything by hand because we're still experimenting, we use high nitrogen materials to actually raise the temperature, because, you know, a large pile usually just gets up to temperature without much addition of high nitrogen materials, and here you end up with a lot of high nitrogen materials. So, you know, if you look at our ratios, you know, we don't usually cross more than 11% of high nitrogen materials in the pile. Whilst if you end up with a massive pile from, you know, of cattle bedding, you're at least 30% of high nitrogen materials, especially if it's fresh. And so that's basically your challenge. And then if you are serious about this, then you need to get yourself, you know, a couple of thermometers and people monitoring it, and actually do a good trial of, you know, how much is going to cost you running that that turner enough for those temperatures to be manageable and for the material not to go anaerobic. The one thing that you will find quite a lot across the country is, you know, people will cover the piles, and then they leave them for a few months, and you take the cover off and it's all white, and you just basically produce a lot of actinobacteria. So we haven't done the experiments, but it's something to consider for growing brassicas in the future. But so for your oilseed rape in bulk. But
Daniel, how would that work for cashing the manure? Covering it over and putting some bokashi in there.
I don't have enough experience. Maybe Adam can comment. So there's people who specialize in it in the country, I know. So they basically silage it. Mm-hmm. But if you were to look at it from the perspective of the soil food web, then you need more material, and I think, you know, you get some control that way. So I don't know exactly, you know, how bokashi basically gives you, you know, what sort of benefits you get in the end. I think Adam.
I think at best, what you would do is denitrify it, so it would no longer be a high nitrogen material, and you turn a lot of the free kind of water soluble stuff into bacterial biomass, which is cool. But, you know, you still need higher successional microbes to eat that bacterial biomass to cycle nutrients. Mm-hmm. So somewhere along the line, you still have to do something like a Johnson Sioux. So then, you know, maybe you can also use stuff like EM1, lactobacillus, to drive down E. coli in the pile. If you wanted to just do that, then you could apply the manure to the field and then have something like a Johnson Sioux. In the Johnson Sioux, you could even put some keratin containing material. Then that would select for keratin decomposing fungi. And then you would have keratin decomposing fungi in your inoculant. You would apply that to field, to the remaining manure, which has lots of, you know, lactobacillus and cyanobacteria and yeasts in it. And then you would have a system where you would be dealing with it in the field. If you wanted to make compost, you obviously have to mix in a hell of a lot of wood chip, straw, shredded cardboard, whatever you can get that's carbon-rich material to deal with it that way. And they're kind of two ways you could go about it. I guess in the future, legislation is going to maybe make it hard to spread manure, right? So if there's room in that legislation to remove pathogens from it using lactobacillus, then yeah, that's cool. But maybe you might just have to compost it.
I mean, legislation basically prevents to put out this material in times when there is no plants growing. So it basically stops farmers from spreading it out at the moment, so like in autumn. And so when spring comes and everything starts growing, then they're allowed to spread it. So yeah, you know, there is even the potential basically of spreading that stuff and simply following it with a good extract, because then you capture this nutrient on the field, and you have the nutrient cyclers in the field.
What about adding biochar to that process?
Well,
Would that give the surface area then to hold on to that activity?
I mean, biochar basically is like a home for bacteria again. So you're just basically creating conditions for bacteria to healthily develop.
What about fungal activity? Will they not survive in there?
I don't know. I've not seen, sort of, you know, somebody would need to point papers to me. I've not seen any work done on that.
No, it's only a question.
Maybe if you can like soak it in humics and fulvics, some really, really old compost, something like 2 to 3 years old.
I'm thinking that biochar. I'm thinking of biochar. Basically, there's many benefits obviously to biochar, but I'm just basically thinking if you want to be adding biochar in bulk to that material, why not just basically skip the process of making biochar and add the wood chips, because that's the carbon that the fungi can actually access. Biochar fungi can't really access that material because it's stable carbon, it's mineral carbon.
The reason for the biochar was to hold on to the nutrients within the biochar and then slowly release them, rather to still spread it in the autumn. Basically.
It's the same. Yeah, I mean, you know, there's many solutions, there's many ways to skin the cat. So if you are adding biochar in bulk, you know, I would just basically look into adding raw material, because
I think it will work to keep nutrients in the top layers. But I think the ultimate thing to do that is probably the microbe network, you know.
And
the way I was thinking was feed the cattle biochar to lower the methane. It's already in the map and already doing the job.
Yeah, perfect. That's yeah.
It's an interesting thing, you know. Whoever came up with that idea, basically, it just reminds me of the black carbon tablets I was fed as a kid, basically to basically prevent diarrhea and stuff, you know. So
that's for sure going to eliminate any of the potential negative consequences of spreading biochar, you know, like it absorbing some nutrients and robbing the plants. So, you know, you're passing it through a cow. So that's a good idea, I'd say.
Already full of microbes then. No, I was thinking that you would be adding this onto the big pile. Obviously,
no, it needs to be activated by some way before it's added to the pile, and by feeding it to cattle. I mean, the cattle seem to love it. They're fed ad lib and they're just drawn to it.
I'd love to follow that if you do that. See how it goes.
We're doing that with Kent University, so I mean we soon share the data on that.
Awesome. This is the thing, and it's like I like this place. You know, this is an opportunity to learn from everybody.
So
how are you making the biochar? Are you making that in-house?
We have been buying it in or receiving it with the grant we just won. We're going to have a biochar retort ourselves.
Okay, is that quite a difficult process to get right?
It's very similar. Everybody says that biochar is very difficult to make, and the more we looked into it, the easier it becomes. It's basically the fines out of charcoal, and it's making 10 mil charcoal. That's basically it.
You could do it with a barrel in the chimney, or you could do it in a hole in the floor with some mud on it.
Mm.
You know, you can even also, you know, in the same process, you can produce an artisan product for artists. You know, if you take some willow, stuff it in the same barrel, basically, you know, do the same process as biochar and don't crush it, and just basically have little chunks like that. You know, you can pack them 10 at a time and sell for 15 quid. You know, there you go, get a business model.
The thing about the biochar, the softwood biochar isn't as good as a mixed hardwood, and chestnut isn't very good at all for making charcoal or biochar out of.
That's interesting.
But a mixed ash is very good. Obviously, there's plenty of that at the moment. Oak, hornbeam, a blend of hardwoods.
What is it based on in terms of, like, why is it better, why is it worse in terms of what the process?
Because each type of wood brings something different to the party.
Mm-hmm. I'm just basically thinking, you know, I'm trying to stop myself from doing dad jokes about sort of the quality of wine in you know, sort of comparing that to biochar, but
never stop doing dad jokes, Daniel.
I will never stop, I promise. I'm sorry, you guys will have to bear with me on that. So yeah, it's interesting, you know, because obviously all of these woods will have different structure, right? Different cell structure as well, and so they all will work differently for the microbes, I'm sure. But does the process differ at all when you're making it?
No, it's exactly the same.
Okay.
It's basically burning without oxygen, isn't it?
Yeah, you're just taking off all the volatile substances.
Mm.
Cool.
Yeah, the trick will be making biochar out of green waste.
Out of green waste. Good luck with that, because you've got lots of leaves in it.
Mm.
You'd probably better off leaving that to us to make compost.
The trouble is, applying it to land is the amount of plastic that's in it.
That's the problem. That's the problem for green waste composters. They have to solve that. And you know, they've been talking about this for years. And I hope and pray that when we come in, we say, you know, like, make some more money by making biology for us, you know, they'll actually clean that up better. I only know of 2 companies that have found solutions, and they basically have to be, they basically had the agency to solve it for themselves. And one is basically saying no to a truck that comes in, and the other one is labor intensive. They basically everybody jumps onto the pile and just pulls out everything. And so
the trouble being, I mean, I've been involved in composting in the past, and it's residual plastic within the pile, and they're just not going to get rid of it.
What do you mean residual plastic in the pile?
Well, what's already on the compost site at the moment from turning and everything else, it's never going to change.
Again, they have to solve it. It's not that it's something. We can't think that there is a problem that doesn't have a solution. It's, you know, why would we be here?
The solution is removing the idiot that puts it in the bin to start with, and you know as well as I do.
But that's the problem. That's the problem. So, like, there's too many idiots along the way. So if you think about this, you know, Robert here, for instance, you know, he's very happy that he doesn't get a lot of municipal stuff coming in. When a truck from the city comes in, basically it's half plastic. And so, you know, whenever somebody comes in with a lot of plastic, he gets rejected, or it just gets charged for putting waste into it, and we try to pull it out. We have done our own sort of analysis, you know, time motion analysis here. So we know sort of how many people hours are required to really pull a good amount of plastic, if not everything. But you need to invest. You need to invest in people. You need to invest in equipment, and that has to be factored into the cost of the material, right? And so, you know, it doesn't matter if you're producing compost of biochar. If you're producing biochar out of plastic, it doesn't really work, does it? So basically, they just have to have good amount of income from the material and some sort of legislative pressure. And there is some pressure, but the environmental agency actually doesn't feel strong enough. And I remember that from a few years ago, so maybe they're building a better case, but they literally don't feel strong enough in themselves to impose those regulations on, actually enforce those regulations on the producers. So they want to go to the public, they want to educate the public, but then it was still before the pandemic, basically. Something else was happening, so they couldn't go to the public with that. And then the pandemic happened, so again, they couldn't go to the public with that, because like, who the hell cares about a bit of plastic in their green bin waste? So they just basically feel that they can't do anything, but they can't justify the extra force, you know, put onto the producers. But again, you know, we're basically going from the other side, and we're hoping to change it from the inside. But yeah, it's a big subject, massive subject.
Do you think we could eventually get to a place where we've got fungus eating plastic?
I hope we get to the place where we don't waste plastic, because plastic is an amazing material. There's you know, the same people that we're working with to produce the biology at scale, they're working with a massive producer of plastic alternative for single use, you know, like the plant-based stuff. And so, you know, they've got a massive project. They got Innovate UK funding last year, and so, you know, they've got like dozen partners, you know, in this whole thing. So they're trying to solve it from that side. And I think, you know, if you're working with material like plastic, which is a fantastic material, fantastic engineering material that's being used because of the plastic industry, because of the packaging industry, it's been forced on us back in the 60s, actually, as a single-use material, because that's how they sold more of it. And when people started noticing that this material is basically polluting the environment, they paid for a marketing campaign of pick it up, and literally just put it on the onus on the society to pick up litter. And that was their solution to the whole thing. They just completely diverted the discussion from the fact that we're using single-use plastics to the fact that we're littering the streets. So it's an unfortunate, you know, turn of events. We're used to this plastic now. So I'm kind of hopeful that that instead of, you know, trying to change, you know, the whole society, we're going to have, you know, a different manufacturing process, different ingredients list, and then the actual plastic, which is useful for engineering, is going to be kept for that. And that's going to be then a product that lasts 1,000 years. You know, it's fine.
I mean, they're also pyrolyzing it to do biodiesel, aren't they?
Please don't call it biodiesel then.
Well, that's not my phrase, Daniel.
It's like a few years ago. Basically, we had this massive PR campaign. It's like I'm in awe of PR, basically, because there was this massive campaign about, so look at Sweden, how, you know, environmentally friendly they are. What do? It's like they got rid of their landfills. How? They burn everything.
Yes.
It's like you just took this fantastic material that can last 1,000 years as an engineering product, and then you made it into a single-use product, and then to make sure that you're environmentally friendly, you burn it so that all of that thing goes into the atmosphere. It's like, yeah, logically. So yeah, stuff like that there. So Ed, sorry, we just got off on a tangent, but these things are all connected. But do you have any more questions? We're drawing to an end, so if anybody's got any more questions before we go off on another tangent.
Well, I'd just like to say, Richard, I'd be interested in your biochar thing and what the results are when you've got a report.
Yeah.
And do sheep eat biochar?
I haven't tried sheep, but I've got a sheep farmer that would be up for the challenge.
Yeah, send a few briquettes to Judith.
Sheep will eat anything.
especially if it's going to kill them.
What sort of amounts are you feeding to your cattle?
It's ad lib, so they just help themselves.
Oh, do they? Just out of a trough, or?
Yeah.
Yeah. Okay.
But you know how much do they go through?
Between 20 and 40 kilos in a pen of 20.
In a pen of 20?
In a week or
in a day. In a day.
Every day?
Well, pretty much. There's some that eat more than others. We got the idea from the Harper Adams trial that's going on, and they're actually mixing it in with molasses to put it in like a feed ration.
And is it quite finely ground, or is it in chunks and they chew it themselves?
Between 10 and 20 mil, we've got it down to.
Right. Okay. Yeah. Mm-hmm.
We're looking at a crusher to get this product. Obviously, the charcoal isn't what we're looking for. We're looking for a more unified product. So if we crush it and produce it like that and apply it to land, and after it's activated, it's just to see what the difference is.
And what's the muck like? You know, what's the dung like when they've cast it?
You can still see the biochar within it.
Yeah. And do you think
because it doesn't break down, just holding on to that nutrients? And I mean, a gram of biochar has 30 square meters of surface area.
Mm. And does it change the consistency of it, or is
no?
that,
It's still crunchy, if you like. It's not. I mean, in its warm form, it can last in the soil thousands of years.
Yeah.
And so we're looking at it from holding on to that nutrients, as well as the benefits that it does in the soil drainage and etc. to improve the soil.
I was just imagining, Richard, you checking the quality and how it's holding the nutrients afterwards.
You joke, Daniel. You've opened a whole can of worms here at Bank Farm. We've got 3 and 4 IBCs now, with composted mix of cattle and hen muck, 2 with worms that we bought in, 1 with worms that we keep on adding to with the Bank Farm.
You're welcome. You're welcome.
And then the 4th one, we keep on adding fungal activity to, either through cattle poo with fungal activity on, or plant pots that we find anywhere, or any kind, whether it's positive fungi or not, we're just adding it.
Buy some goop.
Why not?
Buy some goop. You're gonna have some positive fungal activity.
Well, yeah. Well, you're welcome to donate some, Daniel.
Oh, there you go. Cheapskate.
Did you have your raffle with your goop last night, Daniel?
Yeah, I've got a guy, he hasn't responded yet, so I couldn't send it. It's still in the fridge.
Right. Yeah. Mm-hmm. How often do you do a batch?
Well, we gather sort of from the week and we make it Monday or Tuesday to send it for the next day.
Right. Okay.
If we basically sort of at some point decide that we have to make more, then we'll do it more than once a week. But at the moment we're just starting. We haven't even started advertising beyond this group and the group of the other students of Dr. Ingham. Like literally, the group that sold was a few people here, a few people from my friends group, and somebody else, but that found us sort of along the way. We haven't really started advertising.
Any inquiries from the conference?
Like one liter of goop sold during me talking. So that was nice.
Yeah. Yeah.
So that was nice. So that went out yesterday. No, it's great. It's just like 2 years ago when I was sort of just barely starting off, and sort of anybody was trying to ask me, I was like, well, I've got this thing, how do I do this? Well, let's start with microscopy assay. It's like, and then what? It's like, I can't help you. Now I actually can. So it's good.
Yeah.
So it's nice. We just need to increase the production now. So because that's the next challenge, you know, to actually have enough to do everything. You know me, how ambitious I get, and then I get stopped basically, and just going.
Practicalities.
Reality strikes.
There's so many rabbit holes you can go down with these things, isn't there?
Yeah, main rabbit hole is get more people, more space, more machines. That's the main rabbit hole that I need right now. So that's why, you know, I've got this. That's why I've been basically investing my time and some other people's time in a nice pitch deck, so I can go to rich people and say, you know, cough up, and we can do this properly. So we just want to solve it once and for all. Get it done.
What like Brexit?
And then you can have a garden party.
I'm sorry, Richard. I don't think neither you or me will get invited to that one.
No.
Don't expect that.
No. No.
Talking about conferences, has anybody here been to Groundswell? You know, the conference that they have down, is it near you, Daniel? Or it'll be nearer you than it is me?
Well, it's definitely nearer to me than to you, but it's was in June time, isn't it?
Yeah, I think so. Yeah.
I haven't bought my
Very well worth it. Very good.
It's something I wanted to do a couple of years, but it's just a big thing for me to get down there and find somewhere to stay. But I think it's an ambition for this year. I'm going to try. You
need to basically go for Airbnb in the local area and do it that way.
You can stay there actually. You can camp if you're so inclined. It's a 2-day event, right? But it's you get talking to lots of people in the evening, and it's quite a social. It's a real community thing. It's a good show.
Yeah.
Yeah, it is.
And must make the effort.
It's probably one of the best, if not the best, regenerative ag shows in the UK now, for sure. Because you've got the three main conferences really. You've got the Oxford Farming Conference, you know, the incumbent. You've got the Oxford Real Farming Conference, which was, you know, set in competition, and then suddenly Groundswell came up to say, you know, like, we're just these min-till, no-till people, and regenerative agriculture pretty much latched onto that. And so you're going to get everything from Glyn Mitchell talking about the soil food web to people demonstrating the next best drill.
And is there much about livestock production? Because, I mean, interested though I am in crops to some extent, it's livestock we make our living from.
I'm not going to answer that yes or no, Richard.
Not a great deal. I mean, the universities are the ones for livestock trials. They're doing some really, really good work. Leeds, Harper Adams, Cirencester, they're all doing really good stuff on the livestock front.
It's the grazing stuff that I need to learn. The actual livestock husbandry bit, it's the grazing's our biggest challenge, and relearning how to do that regeneratively.
But I'm just thinking speakers. Hold on one second, because I'm just on the website right now. I'm just going to share my screen. Bear with me. Boom. There we go. Speakers. Anyway, because I mean, there's usually people. Oh, hello, Matt. Jerry, hello. Dominic. This looks like a pasture.
You will pick up information, I'm sure, but it's something that
he'll be well. He does crops as well, doesn't he?
But you need to basically look through the speakers, and that way you'll be able to so judge whether it's worthwhile for you to go down there. There's obviously quite a few here with pictures, even with animals. I would assume, because Niels, who frequents this group as well, he works a lot with pasture farmers. So I would think there's a few people who do manage grazing and stuff like that. There's another one. Look, Animalsies. Charles Dowding even made it there. Look, Adam Driver, we know from this group as well.
Judith, have you come across the sort of Alan Savory Institute
Oh
type of
gosh,
work?
yeah. Me and my dad's carer are trying to read his book at the moment. My God, it's mind bending. It's a lot of reading.
They offer a series of courses, all to do with different aspects of it.
Another one, yeah.
And I've recently done the sort of entry level one, and I found it useful. Right.
So where's that?
It's just online.
Oh, hey, that's not so bad. I could do that.
And yeah, it's pretty good.
Yeah, there's a lot of things. You just need to look at that, make up your own mind.
Yeah.
But last year was
Edward's quite right. It's more than a one-day show.
4,000 people last year, wasn't it? It was massive. It was bigger than any of the other conferences. 4,000 people yesterday in person. So it is the biggest thing for regenerative these days. Cool. Anything else?
All good. Thank you.
Thank you.
Thank you very much for your help with my questions.
Yeah, no worries. Shoot away. Come back next week, and if you have any more, that's what it's called, a drop-in session. Just drop in. All right.
Yep.
Cheers,
Take
everyone.
care, guys. Thank you.
Cheers.
Bye-bye.
Thank you.